There are two other energy sources that our metabolic systems will utilize: lipids and amino acids. We'll explore those two in the 11th lecture in our biochemistry module. We'll start first with lipid digestion and absorption. The mechanical digestion of lipids occurs mainly in the stomach. Salivary amylase and other sorts of enzymes that we find in our mouths is not able to digest these larger molecules. So this kind of digestion is going to have to occur later on. The chemical digestion is facilitated by bile, pancreatic lipase, colipase, and cholesterol esterase in the stomach and the lower intestine. Short-chain fatty acids are absorbed across the intestines into the blood because they're small. However, long-chain fatty acids have to be absorbed as micelles and assembled into chylomicrons to be later released into the lymphatic system for use. Lipid mobilization occurs in two sorts of lipase processes. Hormone-sensitive lipases mobilize lipids from adipocytes. If you remember, adipocytes are the fat cells. Lipoprotein lipase will mobilize lipids from lipoproteins, or proteins that contain lipids within them. To the bottom right is a diagram of pancreatic lipase and what it looks like. As you can see, it has a quaternary structure. There are multiple different ways lipids are transported. Chylomicrons transport from dietary triacylglycerol molecules, and this is done via lymphatic system. As you remember, this is for long chains only. Very low density lipoprotein is similar to that of chylomicrons, but VLDL is produced and assembled in liver cells. Like chylomicrons, the main function of VLDL is the transport of triacylglycerols to other tissues. They also contain fatty acids that are synthesized from excess glucose or retrieved from chylomicron remnants. IDL, or intermediate density lipoproteins, are VLDL remnants in transition between triacylglycerol and cholesterol transport. They pick up cholesterol esters from HDL. LDL is noted as low-density lipoprotein. Although both LDL and HDL are primarily cholesterol particles, the majority of the cholesterol measured in blood is associated with LDL. The normal role of LDL is to deliver cholesterol from to tissues for biosynthesis. However, cholesterol also plays an important role in cell membranes. In addition, bile acids and salts are made from cholesterol in the liver and many other tissues that require cholesterol for steroid hormone synthesis. HDL, or high-density lipoprotein, is synthesized in the liver and intestines and released as dense protein-rich particles into the blood. HDL will contain apolipoproteins used for cholesterol recovery, that is, the cleaning up of excess cholesterol from blood vessels for excretion. HDL also delivers some cholesterol for steroidogenic tissues and transfers necessary apolipoproteins to some other lipoproteins. Oftentimes, HDL is referred to as good cholesterol, whereas LDL is referred to as bad cholesterol. Apoproteins will be the proteins that control interactions between all these lipoproteins as well. Fatty acids and triacylglycerols. These are carboxylic acids, usually with a single long chain or branch chains. If they're saturated, they have no double bonds between carbons. And obviously, if there are double bonds present between carbons, they're going to be deemed unsaturated. How many double bonds are present is going to determine how unsaturated they are. These are synthesized in the cytoplasm from acetyl coenzyme A transported out of the mitochondria, and there are several steps to this. The first step is activation. When fatty acids are first metabolized, they become activated by attaching to coenzyme A, which is catalyzed by fatty acyl coenzyme A synthase. This is referred to as fatty acyl CoA. Later, the bonds will form between those two, and then they will be reduced. After the reduction, they will be dehydrogenated, meaning that water is going to be pulled out, and finally reduced again. And this is going to occur over and over and over again. Here's a diagram of what this type of oxidation and reduction repetition is going to look like. This is often called beta oxidation. It uses cycles of oxidation, hydration, oxidation, and cleavage to break down saturated fatty acids. If they are branched or unsaturated, they will need specific enzymes to proceed. The other type of energy source that you'll often see used is ketone bodies via ketogenesis, which is the process of generating ketone bodies. This only occurs during prolonged states of starvation. It's going to cause an excess of acetyl-CoA in the liver, though. Ketolysis regenerates acetyl-CoA for use in peripheral tissues. Note that the brain can use ketone bodies during prolonged starvation. The brain is our biggest user of energy, so it's going to take most of our resources to keep maintained. The brain often can't use anything else, so lipid bodies cannot be used by the brain. Any sort of lipid byproduct will not be allowed to pass through for use, so the brain often can only use sugar. In absolute desperate moments, it will use ketone bodies, though. Which leads to protein catabolism. This occurs primarily in the small intestines. Catabolism of cellular proteins only occurs under prolonged starvation because proteins are obviously more useful than lipids or carbohydrates, so our body's going to generally want to keep those intact instead of breaking them apart. The carbon skeletons of amino acids are often used for energy as well via gluconeogenesis or ketone bodies. Amino groups are often fed into the urea cycle for excretion after they're removed. Side chains are dealt with according to its type. So again, if you remember, there's 20 different types, and what those side chains are going to dictate what happens to it in the end. Alright, so that was a hard and fast look at lipid and amino acid metabolism. That'll round out our discussion of metabolism. In the final lecture of our biochemistry series, we're going to talk about bioenergetics and the regulation of metabolism. We'll see you then.